Hello, in this video I'm going to show how I made a very cheap gel electrophoresis setup and the troubleshooting I went through to get it to work. My first goal for this project was just to get some DNA ladder and run it through the gel and get some visible DNA bands without focusing on the quality of the bands. I based my design of the gel box off of a video from the Thought Emporium. He showed how to make a homemade gel electrophoresis setup but wasn't able to get it to work with the cheapest supplies. Since in my experiment I wasn't too focused on the quality of the DNA bands, I was able to go cheap. So for the gel box, I just got a little plastic box and stuck 2 graphite electrodes in each end and sealed it with some epoxy. For the gel cast, I used some acrylic that I cut and epoxied together, and then another piece of acrylic that I dremeled grooves out of to make the gel comb. I also needed a way to visualize the DNA, so I built a transilluminator, which was just a box with some blue LED strips in it and an acrylic window at the top for the light to shine through. So for the actual gel electrophoresis, we need to start with a diluted TAE solution. So we just add 490 milliliters of distilled water to a bottle. And we add 10 milliliters of the stock TAE solution to the water. My stock solution was in 50x concentration, so to dilute into 1 liter, you could do 980 milliliters of the water and 20 milliliters of the stock solution. Then we add 50 milliliters of the diluted TAE solution to a flask and then weigh out 1 gram of the agarose. For the first test, I used 1 gram of agarose to make a 2% gel, and then later I tried a 1% gel, which is 0.5 grams of agarose. Then I pulse the solution in the microwave at 10-second increments for around 1 minute and 30 seconds until the solution is perfectly clear. Once the solution has cooled down enough to the point where the bottom of the flask is comfortable enough to touch, I pipette 5 microliters of CyberSafe's DNA stain into the agarose solution. I ran multiple gels with the CyberSafe DNA stain and got no visible DNA. I bought a new DNA stain called Gel Green and also got no results with that. At that point, I just assumed the issue was with the DNA ladder, so I bought new DNA ladder and ran a gel with that with the Gel Green DNA stain and got very bright DNA bands. I will run another gel using the CyberSafe DNA stain and the new DNA ladder to see if the stain was part of the issue. If the CyberSafe ends up working, that would mean that this project could have been a lot cheaper because the CyberSafe was available in 80 microliters, which was only around $40, whereas the Gel Green was only available in 150 microliters, which was around $80. For the gel cast, I just put Scotch tape on each end to seal it, and also so I can remove it and slide the gel out. I take the tape off of the gel cast and slide the gel into the gel box. I pour the diluted TAE running buffer over the gel until it covers both electrodes completely. Then I pipetted 8 microliters of 100 base pair DNA ladder into the first well. In my other tests, I used 5 microliters of DNA ladder, which should have been enough to get visible DNA, but I wasn't able to see anything, so I decided to just try a higher volume of DNA ladder. I used a safe DNA stain as opposed to ethidium bromide, which is typically used to stain DNA for gel electrophoresis. Ethidium bromide is very mutagenic and can be dangerous to work with, which is why I used a safe, non-mutagenic DNA stain. The issue with the safe DNA stains is that they can be less sensitive, which is why I was hoping adding more DNA would fix my problem. It turned out that the quantity or the DNA stain had nothing to do with it, and it was just the DNA ladder. Then I pipetted 3 microliters of loading dye by itself into the last 3 lanes to help track the migration of the DNA through the gel. The general rule is to use 1 volt per millimeter of gel. I used a 60 millimeter gel as was done in the Thought Emporium video. I was able to get decent separation of the DNA using half the voltage and twice the amount of time. It took around 2 hours and 20 minutes. In this gel, the DNA ladder was in wells 1 and 4. These are the results of the gel on the transilluminator. You can see bands of DNA that are matching in both lanes. On camera, the bands are very difficult to see, but in person, the bands are very sharp and bright. In order to be able to see the bands of DNA, I used laser goggles that block the general wavelength of light that the LEDs emit without blocking the wavelength of light that the DNA stain emits. When I change the picture to black and white and adjust the contrast, the bands are more easy to see. You could see that the DNA ladder starts at the bottom at 100 base pairs and goes up each band in 100 base pair increments all the way to 1000 base pairs. Using this, we can identify which band corresponds to which DNA fragment size. Since this picture is upside down, it's labeled in reverse with 100 starting at the top. I plan to make a second video in which I try to improve the quality of the DNA bands, get better separation of the DNA bands, and get better images of the DNA. Some ways I can achieve these goals is by using an appropriate voltage, by using a DC to DC up converter, which is about $30 with my desktop power supply to get to 60 volts. I could build a new transilluminator that shines the blue LEDs into the side of the gel rather than underneath, as to not get in the way of the picture. I could also use a camera that has an adjustable exposure so I can take a picture with a long exposure. I could use post-staining as opposed to adding the DNA directly to the agarose solution. Adding a small amount of DNA stain to the running buffer could also prevent smearing of the DNA.